Today we are detecting DNA using fluorescent silver nanoclusters. My name is Gabrielle Taylor. I'm a mechanical engineering student at Sacramento City College.
And I'm Clarissa Howe. I'm a mechanical engineering student at Santa Barbara City College.
My name is Calvin Peng. I'm a chemistry major at San Joaquin Delta College.
My name is Greg Doty. I'm a biochemistry major at Ventura College.
And we'd like to give thanks to, we worked in the mechanical engineering department with our mentor, Travis O'Donnell, and in the advisor of Samita Penather. Silver nanoclusters is a novel new technology to detect DNA. DNA detection is very important and has many applications when it comes to medical diagnostics, as well as research prominently in the biology area. We want to make it fast and affordable and therefore accessible all around the world, especially in third world countries where often they need the most help. So here we can take a look on how the nanoclusters are formed. So first we will need a DNA probe, and we throw it into a solution with a silver salt and our target DNA. So you only want to find one DNA at a time, of course. Then we throw in a reducing agent. This breaks apart the silver salt and allows the silver to free float and form the silver nanoclusters when the target DNA is present. And so you can see here is some of our test results, and there is in fact fluorescence. So we'll take now a closer look at the DNA probe. There are three parts to the DNA probes. It is made of a synthetic strand of DNA. So first there's the silver nanocluster region. You can see the red region there. And that is the region that the nanoclusters will actually form at. The second region is the probe region. And that is the region that has a very specific sequence of DNA that we'll want to attach to the DNA that we're looking for. And the final region is the blocking region that perfectly hybridizes with the nanocluster region when the target DNA is not present, and therefore blocking any nanoclusters from forming. You can see here on our graph that there is in fact a target DNA, and so it will want to connect to the probe region, so it forces it open, breaking apart the blocking region and the silver nanocluster region, therefore allowing silver nanoclusters to form. So we throw in the silver salt and the reducing agent, and in fact, the silver nanoclusters form. The reason why these nanoclusters fluoresce is because they're so small, only 10 to 100 atoms large, they retain some of their molecular properties, and one of these is fluorescence.
In the Penather lab, we were working with hepatitis B and C DNA. Our experiment was divided into two parts. In the first, we were testing our probes to make sure they worked and to measure their initial fluorescence. In the second part, we looked at aspects of our reaction that we could potentially change to optimize the fluorescence. We combined the parts of our reaction into small vials, adding sodium phosphate as our first solution, the silver salt, the reducing agent, and our probe, either the hepatitis B or C. And then in one vial we added the DNA corresponding to the probe, and another vial we didn't add any DNA. We wanted to compare to see what the fluorescence that we would achieve from the hepatitis DNA and probe combo would be. And then in the other vial we hoped that we'd find little to no fluorescence.
After we ran this experiment, we put our results into a fluorometer, which measures the intensity, which is the intensity of the light emitted of the fluorescence, which is on the y-axis, over specific wavelengths, which is on the x-axis here. Our graph shows our two trials. The first one was the solution with the targeted DNA in it for hepatitis B, and the blue line represents the trial with no target DNA. They both fluoresced around 625 nanometers, which is pinkish fluorescent color. And these are the highest peaks too of our lines. The importance of the difference between the two peaks shows us that we successfully synthesized nanoclusters and that they binded with the DNA probes and fluoresced. We also ran hepatitis C DNA experiment and got similar results. The difference between the peaks with the hepatitis C DNA shows us that those two also worked.
these, and
Oh, sorry. Didn't mean to switch. And these highest intensity peaks for each of them became our standards for our future experiments.
so with this initial intensity in mind, we looked at our reaction and saw different places that we could potentially change the reaction and create better fluorescence. These were the silver salt and the solution. The first change we made was to compare the solution and the chemical compounds of that. We mixed 4 solutions to compare to our original sodium phosphate that had the same pH and the same concentration. In this bar graph, we compared the intensity of our original sodium phosphate result, taking only the reading of intensity at that peak 625 nanometer wavelength, and compared it to our other solutions. You can see that none of them really matched up to our first sodium phosphate. So we knew from this that in our future optimization experiments, we should use the sodium phosphate as our solution. But we looked at other aspects that we could change as well, which is the silver salt. We increased the concentration of silver salt by 33% in our second trial to see if increasing the silver content during the reaction would create a better or more fluorescent silver nanocluster.
What we found out when we ran this second experiment was that the addition of the silver salt, or additional silver salt, actually did increase the intensity of the fluorescence when compared to our standard test that we initially ran. So what we decided was we would take this new information with the additional silver salt and see what other parameters we could change to increase or optimize the intensity. We decided to go with changing the concentration of our sodium phosphate solution to see how that would affect it. So we kept the additional silver salt standard that we ran in the previous test and changed the concentration. We compared it and lowered the concentrations. What we found was that when we increased the concentration of sodium phosphate, it actually decreased the intensity of the fluorescence. And when we decreased the concentration of the sodium phosphate, it increased the fluorescence, or the intensity of the fluorescence. We also ran these exact same trials with our hepatitis C DNA. And none of those trials gave us any more optimized results. All of them were not as good as our initial test under our initial conditions.
The trials we ran on our hepatitis B and C probes gave very different varied results. The hepatitis B and C viruses are slightly similar, but they differ in the structure of their DNA sequence. This means that 2 types of DNA will have different corresponding probes. This explains the variations in our results, even if the DNA probes are in the same optimal conditions. Now, not every variable condition had significantly different results. This is because the hepatitis B and hepatitis C DNA probes worked best in the sodium phosphate solution. Now, in conclusion, we created fluorescent silver nanoclusters that successfully detected the presence of the hepatitis B and hepatitis C DNA. We also did additional test runs to optimize the synthesis of our silver nanoclusters so that we can hopefully improve on our detection methods and increase the sensitivity of our method. Now, in our future goal, we would like to make our method more affordable and quicker to be applied in disease diagnostics. We would like to thank our mentor, Travis O'Donnell, our faculty advisor, the Cooke Bridges staff, the Jack Kent Cooke Foundation for allowing us to participate in this program, and we would also like to thank all of you for listening to our presentation.